Well, thank you very much for introducing the session in that way. And I should say that it's a great honor for me to be here, especially in the context of a forum about the innovative city and in the context of a panel about new metabolism. I believe that buildings and cities are and always have been living organisms. So I certainly agree with and I'm inspired by Nicholas Negroponte when he says that buildings are computers we live in. And I would also like to add that perhaps buildings are biological organisms that we live in. They have metabolisms and they're situated in complex ecosystems with interconnected loops of ideas, technologies, culture, politics, humans, non-humans, and the natural environment. And they're situated in this kind of ecosystem where each loop relates to the other loops. For example, technology would not make sense alone without culture. Environment without justice would not make sense. Humans without non-humans would not make sense. Each project and our kind of collective projects, my collective projects as an architect and designer, but I think also our collective projects as a kind of design community, and as the city, really, our collective projects only make sense through their interconnections. So it's in this context that over the past few years, my practice has been exploring prototypes of this kind of new metabolism through a variety of digital technologies. We've created things like this, a glass that breathes in response to people, combining sensors and information. This is a project called Living City, commissioned by the Van Allen Institute and the I-Beam Center for Art and Technology. We've created a facade that glows and blinks in response to things like air quality and human interest in the environment. Here we're combining environment with public space, and this is a project called Living City in Seoul, South Korea, commissioned by the city of Seoul. We've also created a project that's a wall that displays real-time social media graffiti. Here we're combining robotics with public debate. In other words, robotics that are not just top-down automation, but robotics that have feedback systems. This is a project called Social Robot in San Francisco for an installation of the Quantified Self group. And we've even created this project here, soup bowls that reveal secret messages in street food, combining street culture and critical discourse. So this is a project called Street Life in Shenzhen, China. More recently, we've been designing with living systems, with actual living systems, combining the digital with the biological. And this is an example where we've created an air filtering building facade with the self-sustaining ecosystem of marine plants and animals. So here we're combining building systems and aesthetics or atmosphere. This is a project called Amphibious Envelope. It's in Chicago right now as part of the current Chicago Architecture Biennial. In a more general sense, we've been creating projects recently that are really, I think, at the intersection of architecture, biology, and computation, combining all of them, thinking of these loops as interrelated and connected. But of course, it's important for me to note, as Oren Katz described in a slightly different way, that architects and designers have been fascinated by biology for many years. Here's a book that was written about 100 years ago that continues to be an inspiration for architects. But I'd like to say that biology of today is very different than 100 years ago. It's now possible to do things like this. We can grow cells in glass chips outside of a living organism. We can do things like this through quantum dots, show the neurons firing in real time in a living tadpole. We can monitor the molecular signaling between individual stem cells in an organism that's growing and developing, and these molecular signals tell each cell whether to grow into tissue of bone or heart or muscle. And we're able to do things like this: take biology, the growth of bacteria on a petri dish about this size, and then start applying some current techniques of computation to it. So we can use computer vision, we can use machine learning to try to understand and interpret what's happening in this very complex ecosystem, or this complex metabolism of biological growth. And once we can put that complex biological growth into the computer, then it becomes very interesting to me as a potential design ingredient. In other words, I think this new version of biology right now, different than 100 years ago, offers several new possibilities. And what I'd like to do is describe 3 of those possibilities that we have been working with in our studio. Number 1, biocomputation. This basically means collaborating with living organisms to compute solutions and explore designs beyond what is possible for a human alone to create. And in this project, we started with taking microscope photos and videos of xylem cells in the stems of plants and converting those videos and photos into 3D digital models of the growth of xylem cells so that they're in this kind of design environment. And then we take a bunch of different xylem cells that create these exoskeleton shapes, and we start trying to derive data from them. So here we're measuring the length and angle of each bar in the xylem cell, feeding it into a spreadsheet, then using a kind of version of artificial intelligence, this great computer system created at Cornell University, to derive an equation from a very complex data set. So you can take almost any spreadsheet, feed it into the computer, and the computer over time will try to evolve equations that describe the data. Now, why would we want to do that? Well, because then we can start creating xylem cells in the computer, or creating a biological algorithm that describes what the xylem cell does. Then we can recreate some xylem cells that exist in nature according to a straight center line, the line in blue, but then we can also apply this biological algorithm in a new context that never existed in nature. So we can create an L-shaped xylem cell by creating an L-shaped blue center line and try to compute through this biological computation process how complex biological growth would create a form that never existed in nature. So this is one of the ways that we have been trying to explore what is possible through the combination of biology and design. And to make it more practical, we're not just doing this as research, we're doing this as kind of applied research. To make this more practical, we're applying this in an initial case to the design of something like a chair. So what you see here is maybe a representation, a simplified representation of how design might normally occur. You make a sketch, a kind of stick figure of a chair, and then you slowly figure out how to make it in the real world through existing materials and what is available and fabrication methods that exist. But what if we could have a new approach to design where we sketched out some things, like the seat of a chair, that we knew precisely we wanted. But then we sketched out other parts of the chair, for example, the supports of a chair, that we wanted to explore and learn something new about and use a new design method for. And that was the basis for our thinking about this project, where we took the xylem cell algorithm, the biological program for how xylem cells grow, and we applied it to try to discover something new about how the support of a chair might look. So the details are not so important, but we basically populate a series of points, connect these points into a lattice, and we basically have an environment in the computer where we can design or generate many versions of a chair and then simulate their performance, for example, structural performance of a chair, and explore thousands of possible designs. Use the computer to do what it's good at, repeating tasks over and over again, and create this data set where each point represents a potential chair, and allow a human to do what they're good at, which is to interpret results, apply judgment, negotiate trade-offs between competing objectives. And in the end of the day, we think this is a promising approach for using biocomputation to create new kinds of objects in the physical world that would not necessarily occur to us, that would not be possible to design with other methods, with previous methods. Number 2, biosensing. So for us, this basically means using living biological organisms to detect information about the environment and then respond accordingly. This is a project that we created and that we're building right now for the city of New York. And it starts with the idea that we could create a public interface, a real-time interface to environmental quality in a very urban space right in downtown Manhattan. Here's an early version of the project that involves this network of floating lights that tell us things about the environment. And more specifically, this is built with an array of floating tubes. Each tube is about 2 meters long, half below water with sensors below water, half above water with dynamic lights above water. And we have been using, for several years in the early prototypes, some digital technologies. You know, these kinds of physical sensors and microcontrollers that are making up what we talk about when we say the Internet of Things. And we're using this kind of technology in our public installation, but we've also started using biological sensors. And what I mean by that is that we're using mussels, living mussels, the shellfish. And here you see how mussels, when they live, they open and close their shells as part of their process of living. So this is a sped up video, and you can see the pulsing and the moving of the mussels in this tank as they're living. Well, it turns out that the rate and the amount that mussels open and close their shells is a very sensitive and sophisticated detector of pollution in the water. In fact, when mussels open and close their shells, they tell us more about water quality than our most expensive and most sophisticated single-purpose digital sensors. They're a better sensor of water quality than a $10,000 dissolved oxygen sensor. And so the project that we have been doing involves taking living mussels, gluing to one side of a mussel's shell a $2 Hall effect sensor, gluing to the other side of the shell an inexpensive magnet, and then figuring out that we can harness the natural ability of these mussels to detect water quality and put that into a kind of ecosystem of design. Use that biological sensing as part of our design palette, the way we used other digital technologies or fixed inert materials. And we think this is an interesting approach because you could say that it combines artificial intelligence with biological intelligence or natural intelligence. You know, so artificial intelligence is kind of the best computers have to offer, the holy grail of computing. But maybe we could get more out of it by combining it with a version of biological intelligence. What mussels, living mussels, have evolved to do over millions of years, detect a huge variety of chemicals and molecules in their environment and react through a combination of factors. What we're thinking is that wouldn't this be an even better solution than either one alone? So artificial intelligence has great things to offer designers. Natural intelligence has great things to offer. And if you can combine them, that represents what we think is an explosion of new possibilities for future cities and buildings. This project, of course, is not just about an exploration of technology, but it's out in the city. And here is a version of a prototype that we created showing how our dynamic lights, which change color to indicate relative water quality, how those lights look in the context of the skylines of our cities. So this is displaying light as information to the public as a real-time detector of water quality. And we're using this technology not just as a kind of science experiment, not just as a design experiment, but in the context of kind of noting and declaring a new kind of public space in the city. So this is a rendering of what our project will look like after it completes construction in the summer of 2016. It's a 200-foot-long floating pier of these interactive lights that will tell the public things about their environment through this combination of digital sensors and biological sensors. This is in Manhattan in the East River, right in downtown New York City. And number 3, and this is my last category here, this is called biomanufacturing. And that basically is what we're defining as collaborating with living organisms as tiny factories to produce the physical stuff of the built environment, to produce building blocks or even whole physical systems for new kinds of buildings and cities. This project was something we created for the Museum of Modern Art and MoMA PS1 in New York City. And it's a project that began with a competition. So this is our competition video that described the idea we had for biomanufacturing. So this project is called Hi-Fi, and it was basically an experiment in whether we could manufacture a new kind of building block through a new kind of metabolism. So we started with a loop of our own. I think maybe everyone will have loops in this session to show some of their ideas about metabolism. And the loop we're showing here is the carbon cycle, the Earth's normal, natural, healthy cycle of growth and decay and regrowth. And the idea that we had for this project was that maybe we could make our buildings and our cities by temporarily borrowing material and energy from the healthy carbon cycle and then returning it back to the carbon cycle. In other words, we were interested in thinking that maybe we could take low-value raw materials, the waste shown in the diagram, take low-value raw materials rather than high-value raw materials, spend almost no energy converting the raw materials into building blocks instead of spending a huge amount of energy to create building blocks as we do with most buildings. Then create some kind of building, and at the end of the useful life of that building, taking all of that physical stuff and returning it back to the carbon cycle instead of leaving that physical stuff in a landfill for 100 or even 1,000 years. So how could we possibly do this? Well, of course, through a living organism. This is the growth of mycelium, which is the root-like structure of mushrooms. So it grows in thin white filaments that branch in a kind of complex biological algorithm of their own. All of the nutrients that flow through these organisms are part of its growth, but also of the kind of composting of the floor of the forest where these organisms grow. And we were interested in taking this organism, mycelium, and using it to create building blocks, because it turns out that you can combine mycelium with agricultural waste, so not the high-value part of agriculture, not corn kernels, but the low-value part of agriculture, the chopped-up corn stalks waste of agriculture. You can combine those two in a mold of almost any shape, and here you see in about 5 days it will grow into a solid object. So you're able to create useful physical objects out of waste plus a living, growing organism. And our idea was to create a new kind of brick, a new kind of architectural building block through this process. Here's the brick that we designed and that we ended up using for the project. But it's not quite as easy as just describing what the idea is and making sure that it works for one brick. And because no one had ever built a large-scale outdoor architecture out of this material, we had to do a huge amount of testing. So here we are taking one of these bricks, crushing it through a universal testing machine in a laboratory at Columbia University, and also testing an assembly of bricks in this testing machine in order to get information about this new kind of material. In other words, when we took a new idea and a new approach to metabolism out into the world, we had to do a huge amount of testing. And that wasn't just to learn about the material, but it was because if we wanted to create a large-scale installation out of this material, we had to, of course, do some structural analysis. And our structural engineers at Arup, the global engineering firm, said the first thing we need to do is calculate how this will perform. But of course, in most structural engineering software, there's not a drop-down menu item for mushroom brick. So we had to input the data, we had to derive the data and then input the data about how to calculate the performance of our structure. Another challenge we had was that this material is different on the outside than the inside. In other words, if you cut these bricks, the inside is a little bit more porous. And this created an interesting challenge for us because unlike a traditional brick, which you could cut on site in order to make a complex form, we were not able to cut our bricks. That would have compromised their structural performance. So here we had to engage computation to solve two problems that are easy to describe but hard to solve. One is a fitting problem. So if each course of bricks, each line of bricks is a different length, then you have to figure out how you can fit each course of bricks in a row. And also, we had a stacking problem because you have to make sure that each brick alone sits on top of two other bricks and is connected to make a system. And the fitting problem and the stacking problem is something that was impossible to design and understand by hand. We had to use a computer to do it. We wrote a few different algorithms. The writing of the algorithms was iterative. The first one didn't work, but we kept working on it. And here we were applying computation not as the driving force of a design, but to solve a very specific problem in the design process. This project has a very short time span. We had about 3 weeks to construct our structure, and we had solved a lot of the problems, but not all of the problems before we went into construction. And something interesting here about our ecosystem of design, or our metabolism of design, is that we had to combine different forms of knowledge. We had to combine the knowledge of Columbia University graduate students in architecture who know a lot about geometry and computers, and the knowledge of New York City brick masons who know a lot about stacking things. Neither one alone knew enough to solve all of the challenges of this new shape and this new material, but together they were able to collaborate. And maybe it's an example of the kind of heterogeneous collaborative teams that Nicholas Negroponte was describing. Here is a photo of the completed structure. In a way, it was a test of scale. So this is a 13-meter tall structure with 10,000 bricks to test out, could we really make something like architecture out of this new material and this new metabolism. And here you see the structure in the context of the way we typically make things, in the context of the glass and steel buildings of Manhattan, and in the context of the traditional brick buildings of Queens and MoMA PS1. The project in many ways was a test of technical things, the technical performance of building materials, but it was also, in a way, a test of aesthetics or atmosphere, a test of architectural quality. So we were very curious, what would it be like to stand inside this type of construction? What would be the qualities of light and shadow, pattern and texture? And here you see it kind of framing the natural environment, having a different appearance in the foreground and then in the background. But of course, anyone who has been to MoMA PS1 for an installation like this, anyone who's been there knows the ultimate test for this kind of project is its ability to host a party. And here was the thrilling but also terrifying moment when 5,000 people come into the courtyard of MoMA PS1 to hear experimental electronic music, and they're climbing on our structure and going inside our structure and pulling at our structure. But that was entirely fitting for us because in the end, in the way that we think of architecture as being part of an ecosystem, we wanted to test this project out in culture, in society, not simply on a lab bench, not simply in a vacant lot the way some architectural facade mock-ups are done, but out in culture, out in society, out in the context of some people who have very different thoughts about the experience and the construction. Of course, the project also was engaged through a kind of conversation in social media. But really the ultimate test of the project from, the original idea of the project was whether we could take this physical stuff, whether we could deconstruct the building after the end of the summer, break up the bricks into small pieces, combine those small pieces with soil and food scraps and bacteria in a composting facility, and then return all of that physical stuff to soil that in turn could grow new plants or new crops. And we returned all of this soil. We took all of the 10,000 bricks, composted them, returned this soil to tree planting in New York City and local community gardens. And if anything, the big idea of the project is that we might think about the built environment and the buildings and cities of the future in terms of designing them to disappear just as much as we design them to appear. In other words, designing them to return back to some kind of metabolism or cycle. Thank you.